young faculty and stuff like that. So what I'm going to do is talk about this center, why we created it, what its mission is, and sort of then I'm just going to give you snippets of our graduate students' research, some of the things that we've done so you get a sense of the breadth of the kinds of things we're doing. Before I do that, I just want to say I know a lot of you have collaborators at FIU. There's a long-term collaboration among a bunch of you. But we've changed some things. When I got there, I came as the director of the South Research Center, and a lot of you know that for all the Everglades research and restoration that's gone on, and including being the center where the Florida Coastal Everglades program is. But we had all these other water people, like these marine weirdos, you know, that were sitting over in different labs and stuff, not really communicating with each other. We had people in engineering doing sort of water stuff. And so one of the things we wanted to do was to sort of pull all the water science related people together in one place so that we could enhance communication, enhance collaboration. Most of the new grant opportunities are interdisciplinary, and so we've built the Institute for Water and Environment where we brought together freshwater people, the coastal people, and the marine people. I think it's a little bit unique to have that big group of folks all in one institute where we all know each other and talk to each other every single day. So we now have 102 faculty members across virtually every college in 14 departments that are part of our Institute for Water and Environment. This is what it looks like. You can find this on the website, but this was the original sort of center where all the Everglades restoration work was, and all the water quality chemistry and all the stuff. A lot of you know CERC. It's been around for a long time. About 2 months after Rita and I arrived, the president of the university said, hey guys, you need to create a sea level solutions center, right? So that was our first job, even though I was the director of this. And so I wondered about that. It's like, I come to a state where the governor says you cannot use the terms climate change. My president calls me in and says, I want you to create a sea level solution center. It's like, this is a clear case of let's let the new guy do it. If he gets fired, who cares? He hasn't even bought a house yet, right? But Rita and I did. We slogged away and we actually got this thing approved by the Florida Board of Governors, believe it or not. And so we have an official center. We are in the throes, Rita's chairing a committee right now to look for a director. And it's an open until filled. So if anybody is interested in sort of a high level, high profile, under the governor's thumb position, we are looking for a director as we speak. And then we brought all of the marine scientists together under our Marine Education and Research Initiative. We are soon to put in a proposal to the governor's office, again, the BOG, to formalize this as a Marine Education Research Center. So they'll also have state center status. Okay, that's where the Aquarius program is and stuff like that. And then we have cross-cutting programs. We have socio-ecological systems people. Those are mostly social scientists. We have an international water program. We have a lot of AID money. And a lot of projects across the world. And then we have this really fascinating group of people called the Sustainable Built Environment and Informatics folks. They're architects, they're lawyers, they're computer scientists, they're engineers, they're ecologists, and they're interested in envisioning what the new world looks like from a sustainability perspective. So it's a really eclectic group of people. So all of this makes up the new Institute for Water and Environment. And then what I'm here to talk about, Deal mentioned, just recently we convinced NSF to give us money to create the Center for Aquatic Chemistry. So this is what a CREST center is. This is from NSF. If you want one of these, you have to do these things, right? Integrate education and research, new knowledge, diversity, et cetera, promote faculty engagement, and if you're successful, these are the things you do, right? So that's basically what our eyes are on. We obviously got the first part right, and now it's the products, all the students and their success that we really focus on for the next 5 to 10 years. These are 5-year grants, but they're typically renewed unless I do something really stupid. Okay, so this is our mission: detect the sources, transport, transformation, and ecosystem responses to contaminants, pollutants, and natural stressors under changing land use and environmental conditions. A lot of you guys are working on the same thing, thinking about the same problems. Of course, we're situated in an interesting place. We're in Miami. We got the Everglades north of us. We've got all these bays. You've got a similar kind of a thing. So as I said, we have 14 departments, 4 colleges, 2 centers, and we have a STEM transformation institute that's all part of this center and was part of it from the very get-go. So we're trying to understand how to understand chemical contaminants, toxicants, etc. So the context is in South Florida, and you guys have similar things. We've got this really patchy mosaic of land uses, right? So we've got the Everglades that we all know about and most people understand the importance. Then we have one of the most densely populated coastal zones in the world, and in fact the most vulnerable coastal city in the world in terms of the amount of resources that could potentially get lost. And then we have a big agricultural place that's sort of in between all of this stuff. And so what we said was, what we need to do is to understand how these transitions, you know, stuff moving from place to place, what the things are that are in it, how they get transformed, how they get moved, and ultimately what the impact is to ecosystems and down the road to human health. And we tie this all together with the energy, food, water nexus. And basically we know that we depend on the Everglades for our fresh water. The urban folks, all the agriculture stuff depends on the same water for irrigation and agricultural production. But we also have to use that water as a source for energy. That comes with particular costs in terms of the urbanization and the things that humans put out. And then there's also costs and interactions with the agricultural system, right? So all those things sort of cycle together, and to really solve problems and understand what's happening, we have to think about it like this, okay? So we've got sort of 3 focus research areas in the center. We have advanced sensing and environmental exposure, contaminants and pollutants. How do you measure the stuff? How do you identify the stuff? What is it? We want to know, once we know what it is, where does it go? Does it transform as it goes? And how does it get there? And then ultimately, what is the impact? What does it do to ecosystems, communities? What might it do to humans? But another really important part is, this is complex, right? There are a lot of complex interactions. There are a lot of really big, complicated data sets that come out of this kind of research. So one of the big parts of this is, how do we use data analytics, and especially state-of-the-art visualization techniques, to take really big, disparate, complicated data sets and make them available and understandable to the people who matter most, right? The people who actually live in the landscape. And then ultimately we want to do something useful for policymakers and decision makers. And you guys, everyone I've talked to, everything you do certainly informs policy and decision. So this is us in a nutshell. Detect and identify the things, how they move, how they change, and then what their impacts are, and how can we visualize the data, but also the potential impacts from a human perspective. And all of this has to be integrated through discovery-based education, through good education. How do we capture the imagination of high school kids, get them into doing this kind of science, get them to come to college, stay in college, get PhDs, and move through and basically enlighten us in the future. And Rita's the director of sort of all of this part, so if you have questions about that, I'm sure she's happy to answer those questions. Okay, so our first research focus area, we're trying to look for methods of detection. The key here, the goal is, typically once we measure something, it's too late, right? If you measure a huge plume of phosphorus hitting a bay, it's too late, right? There's nothing we can do about it. The phosphorus, the algae are going to take it up, we're going to have a bloom, and all the ensuing things are going to happen. So one of our issues is, how do we measure stuff at much lower detection levels so that we can start to say, uh-oh, we might have a problem, okay? The other thing is, a lot of the chemicals that we measure are really complicated. Those of you who work in oil know that an oil droplet is 40,000 kinds of molecules, and they change like every second. With every beam of light, oil transforms itself, right? So how do you even know what to measure? We don't know what those things are. So people that are in this first research area are really our chemists, and people trying to define or to come up with methods, including molecular biology techniques to find markers and stuff, to help us provide early warning systems, to help us measure things, to know that we have a problem. This is Paulo, one of our most recent PhDs. He just defended about 2 months ago. He has done some amazing stuff. His dissertation is almost like how to know unknown unknowns, right? So it's like he doesn't even know what these chemicals are after an oil spill that gets into an aquatic system. We don't even know what to look for, you know? If you don't know what the chemicals are, how do you know which probe to pick up, or how do you know what analysis to do, right? So he wants to know, his dissertation was developing methodologies to allow him to go through and characterize stuff that we don't know about. And so he said he modified, I have no idea what he does, right? But I'm trying to tell you from the slides he sent me, he has modified some stuff, and he has the ability now to put a sample of water, and he's been thinking about oil most recently, so I'll keep talking about that. He can take a sample of water, put it through a mass spec that he's modified with some other optics, and so it picks up the things that we know about, but it also picks up some molecules that we don't even have a name, right? There's not a name for these things, but it has a molecular structure. And he uses these analytic techniques then to, it's almost like, it seems to me like it's almost like a PCA. He's got these groupings of things based on their molecular structure. But he can then basically come up with all of these different kinds of molecules and their transformations. And one of the things he comes up with is that he starts with a droplet of oil. It's got 5,200 molecules, and within a very short period of time, I mean, these are hours that are shocked with different kinds of light, he ends up with 47,000 unique molecules, right? I mean, that's the kind of stuff. He just published a paper in PNAS. So this is the kind of methodology that we're looking for. How we end up naming this stuff, I have no idea, but at least we know we can find stuff, and oil's a big thing. This is the newest thing. This is actually an undergraduate from Puerto Rico. She's working with Piero Gardinali. They're trying to come up with a quick and easy method where you dip something, or you take a water sample, you put it in this incubator, basically this handheld incubator. You illuminate it, and then you basically look at fecal coliform production, right? Then you use a phone, and you take a picture of it, and it sort of gives you a data set that then you send to a general database. If this works, we don't know if this works, but if we can get it to work, it'll be an amazing thing to use for citizen science, right? So people can go along. Fecal coliforms are really of interest right now, especially in Miami Beach, because we have all these big pumps, we've got leaks in the old sewer systems, and so people were worried about, during king tide and stuff, floods, should their kids go swimming, where should they go swimming, where they shouldn't go swimming. So we may be able to collect a whole lot of data if we can develop this kind of methodology. Abe Smith is working at the biological side of how do we detect things. So he asked the question, and we had this conversation with some other folks here. After something like the Deep Horizon oil spill, everybody goes to the standard EPA ecotoxicology studies. We use fathead minnows that aren't found in the Everglades. We use, the standard fish, and we put them through the standard drill, and we say, oh, geez, how toxic is this stuff? Abe said, let's think about what happens to the native organisms and the organisms that you wouldn't typically think about. So he looked at a whole plethora of organisms to ask the question, how toxic are things? What's the level of toxicity that we should start working on? And so I think what the most important thing from Abe's stuff is he gets these standard toxicity curves that everybody usually uses for one species, but he runs these for all of these native species. And what he finds out is that the toxicity for one thing isn't the same as the toxicity for another thing, right? Well, that makes sense. But now what he's created is a list of species. If you're not too worried about, if you're happy enough to have a high level, you can use these species. Just make sure you don't go through. If you're really worried about a system that is very sensitive, you should use these species, because they'll tell you something at very, very low concentrations, right? And they're the native species that are impacted. The second focus area is we've got all these transition zones going from ag to natural, natural to urban, and back and forth. how does this stuff get there? What happens as it's being transported? How does it change as it moves through the system? And so this is really the hydrologic part of things coupled with our biogeochemical cycles, right? And some chemistry in terms of transformation. And so we've got students that are sort of looking at, this is an agricultural area. These are all the canals that sort of drain Everglades to this side, but all this agricultural runoff ends up in these canals and ends up in the bay. And so the question is, you know, what are we really transporting to the bay, to Florida Bay, and are there better ways of thinking about agriculture and how we manage our canal systems? This is one of the newest students. Her question is sort of a broad question. We know we are doing all this stuff with Everglades restoration. We know we're moving a huge amount of water through canals. We're trying to treat water in various ways, and the question is, are we successful? How can we be more successful? Of course, those of you who know the Everglades story, phosphorus is really the key issue. If phosphorus goes too high, we lose the algal mats that we have, and the whole ecosystem sort of goes through a phase shift. Again, for those of you who work in the Everglades, I apologize, but we know that historically this is what the Everglades looked like. We had this sheet flow coming down, going into Florida Bay. Then at its worst case, we decreased it a huge amount so that now we divert the flow to the east and the west and minimize the flow to the south, so we completely alter the hydrology. And now we're trying to fix that, right? All the billions of dollars that we're spending trying to basically restore some of that sheet flow, trying to restore the directionality and minimize some of the impacts that we've had on the coastal systems. So that's the goal, and we can argue about how successful we have been. Part of the Everglades restoration, of course, has all of these massive large-scale projects associated with moving water, cleaning water, getting the hydrology right. One of the ones that seems to be working fairly well are these storm treatment areas, right? So one of the big issues, like after Irma, you get rainfall events, the reservoirs fill up, the canals fill up, people get worried about dam breaks, breaches, and so they release water. Well, that water from the agricultural areas is, of course, really high in phosphorus. If that breaches and goes into the Everglades with high phosphorus, we get really serious ecosystem responses. So these storm treatment areas are meant to basically be like filters, biofilters, right? They're artificial wetlands. The water is pumped into those. The macrophytes take up a lot of the phosphorus and nitrogen. Some of it gets oxidized, and so then after they get sitting for a while, that water can get released, and it's more like low nitrogen, low phosphorus content water that can then go into the Everglades, okay? So the question is, so it's a small scale fix for a really large scale problem. But before we invest the next 10 billion or whatever it's going to be, we need to know, is this actually working? Are we successful in basically rehabilitating the water, so to speak, in order to get clean water and more water into the Everglades? So what Asefa's group is doing is they're basically taking a modeling approach. They're modeling what used to be the case in terms of surface water depths and flows, what is the current case, and then doing a whole series of simulation models. If we manage the water like this, if we send it through the STAs like this, if we hold it for this long, what will the ultimate hydrology look like? And how can we maximize the amount of water that we can get from the north to the south in the Everglades, okay? But it's not just the amount of water. A lot of bad water, a lot of water with high phosphorus is worse, right, than low water. And so coupled with the hydrologic modeling then is the modeling of what's going on with phosphorus content. So this is probably what it once looked like. This is what it sort of currently looks like. And right now the results aren't horribly great. If we keep doing what we're doing, if we keep running the STAs as best we can, we get some increase in, well, some decrease in phosphorus, increase in water quality in some places, but have these big hot spots. And so, you know, these models are really important in informing the Southwest Water Management District, USGS, and all those management agencies trying their hardest to fix this problem in terms of sort of what's the next step. Do we continue doing this, or do we sort of change? And so we've got a bunch of labs and a bunch of CREST students who are working on sort of this aspect of the fate and transport of particularly phosphorus. Okay, our third area then is sort of that synthesis area. So what? So what if high phosphorus gets down to the Everglades? So what if copper moves from the agricultural systems down to the mangroves of Biscayne Bay? So what if you know, the agricultural runoff ends up in the bay in South Florida? And how do we even say so what? How do we even ask that question or get people to think about that question? How would they visualize it? Okay, so that's the idea behind our third research area, is to sort of do the risk analysis, make predictive models, try to inform people about what the risks and importance of this is going to be, but then also try to come up with better ways of harvesting really dense, complicated data and making it palatable and understandable for the people who actually matter. This is one of the most unique PhDs I've ever funded. Just a PhD student in computing sciences, and what he's interested in is coming up with a better Google, right? So he wants to come up with a way where a PhD student can sit down and ask a question about something and get all of the answers with a single query. And I'll explain that for a minute. So they've created this sort of environmental ontogeny program, okay? So it's a semantic search engine. So a PhD student walks into CREST and says, I'm interested in contaminants in Everglades sediments. Okay, seems simple enough. How many contaminants do you think? How many ways do you think there are as sediments? Could be cores, could be sedimentation, could be sediments, could be clay, could be sand. I mean, you take a single word and think about how many different terms there are out there that really are about sediments, okay? And I know a lot of you think about sediments. So what they do is they create this program in which they create an ontogeny of words. So they basically mine the literature for every term that they can find that really has something to do with sediments. And they build a relational database, right? So now when you type in sediments in this query, it searches for 400 terms that mean sediments, right? All in one shot. Okay, so they've been working on sort of, this is one of the great projects where we have a hydrology PhD student working with a computer science PhD to try to come together to help each other. He's developing this program as part of his dissertation. She really does want to know about contaminants and sediments in the Everglades, okay? And so it's a painstaking approach, but it's basically like a training approach. You start with 5 papers and you have to sort of manually build these ontogenies, and then you start adding papers, and pretty soon the program itself goes into papers and starts harvesting the words. Okay, so it's really an ingenious thing. It's different than anything I've ever thought about. One of the interesting asides is we meet every week in what we call the CREST Journal Club. And we have students talk about different projects, or Rita talks about writing successful proposals or something. So when you submit the CREST proposal, it's a really hard proposal to write. You have the standard 15-page overview of what the CREST Center is going to be, the education, science components, and how they're going to be integrated. But then each of the science sub-projects are a separate 10-page proposal. But then in the end, they have to be tightly linked, right? You have to show how one relates to the other, and they all relate together, and how they all relate back to the core proposal. So he was explaining to us how he was doing this, and we said, oh, this is interesting. Why don't you analyze our proposal? If we did a really good job, we'd have research focus area 1 would have a lot of overlap with 2, and there'd be a lot of overlap in 3, the synthesis part, with 1 and 2, and all of those should have a really lot of overlap with the overview that we wrote, right? So he does this, and we said, so what's the result? And he said, it appears you wrote 4 independent proposals. So, I mean, it's a really fascinating way to think about things, and thank God the NSF reviewers don't have that ability. Another project is one of my students. She's interested in, one of the transitions we have is we have all the citrus farms north of us. And so when we get slugs, when we get a variety of kinds of diseases, the citrus farmers often will spray heavily, even by low-flying airplanes, high amounts of copper. Okay, so copper, it kills snails, but it also kills algae. It does all kinds of things. And then it basically, it comes in spurts, right? So it's not something like agricultural runoff that every time it rains, you get a little bit of phosphorus, you get a little bit of this. This they spray all at once over huge expanses of area, and then the next rainfall, all of that stuff washes into the canals, and because the flows are high, it comes rushing down the canal system, and it hits the estuaries, the mangroves, and it sort of just stops, and it sort of sits there, and it gets taken up. So Tiffany's question is, what does that mean when you get these? They're not very common, they're rare, but they're really large spates and doses of copper. And how does that impact sort of the whole mangrove ecosystem food web? So she's using sailfin mollies as sort of her first case test, because sailfin mollies are found throughout the Everglades and in the coastal zone. They're one of those fish that can live in freshwater and live all the way up to 30 ppt of salinity, right? And so they can bounce back and forth quite easily. And so she's basically taking sort of standard tests to look how copper is impacting them. But what we're really interested in is not the LD50, what dosage is it that they die or stop reproducing. I'm really interested in this lower dosage area. At what levels of copper do they start exhibiting behavioral changes? It's all about predator-prey interactions. If you're a small fish like this, you're prey for all the other fish. If you can't get away, if you can't swim fast enough, you don't have any burst speed, then all of you are going to die. It doesn't matter about reproduction and growth because you're going to be eaten, right? And so we're really interested in that behavioral component of what is copper doing to the lateral line system, their orientation abilities, and their ability to swim. And so she's showing that under not as low doses, thankfully, as it could be, but under intermediate dosages, these guys really do lose their complete orientation. They don't know what's upstream, they don't know what's downstream, and they certainly don't recognize big predators coming at them. And so she's going to build a food web sort of based on these kinds of low dosage responses to copper. And so she's pretty new in her thesis, but some of the preliminary data look pretty good. Now, I showed you this before. These are our research focus areas, okay? When we wrote the first proposal, we were very focused on the Everglades, because that's what most of our experience is. Most of our faculty have done a lot with Everglades. And so a lot of the early work and the whole early proposal was really concentrating on the Everglades and its tie to agriculture and urban environments, okay? One of the great things about something like a Center of Excellence grants is NSF allows you to write supplements every year. So you can get up to 20% of the award every year to do something new if you can convince the reviewer. So 20% of $1 million is 200,000, that ain't bad. The first thing we did was we said, well, you know, all this stuff doesn't stop at the end of the Everglades. We really need to think about that transition system called the mangroves. And besides just trying to understand what toxins, contaminants and stuff that come from upstream and basically pool up around this, we don't understand really very well how much of that stuff is taken up by the mangroves or what it does. And, oh, by the way, mangroves turn out to be one of our most important sentinel systems, maybe, in the face of sea level rise. There's a huge question about whether the sedimentation rates can keep up with the sea level rise rates. And, you know, the jury is still out. In some cases, It seems like mangroves are taking over in the case of Houston and taking over salt marshes. In other cases, the mangroves are retreating because sedimentation rates aren't high enough and sea level rise is too fast, and so we're losing mangroves. And so it's a really important question to try to understand that sedimentation process. And so we were very lucky in our first attempt to get a supplement from NSF to add mangroves as a critical ecosystem to the CREST Center. And that spurred on a whole new set of science questions, including a great new collaboration. Some of you might recognize this guy. This is Donnie Smoke from here, from USF. He's in the other campus, I guess. I don't understand. He's in that building, but I guess you don't talk to him. I don't. Anyhow, so this allowed us to bring in Donnie because Donnie is known. If you talk to anybody in South U.S. or the Caribbean, and they're building models and trying to understand and predict mangroves and whether accretion rates can keep up with sea level rise, they say, oh yeah, we've got these models, here's our predictions, but we have this one uncertainty about this one key parameter. What is that? Well, it's that accretion rate parameter. What are you using? Well, this guy Donnie Smoke created this one thing one time, and everybody around this area uses Donnie's correction factors or sedimentation rates. And so we have this great opportunity to compare South Florida, where we have lots of data and lots of knowledge, with Puerto Rico, which is a really different place, mountainous, different kinds of geology, different kinds of drivers, high densities of people, and sort of start a comparison and collaboration between USF, UPR, and FIU. And so we had 3 sites that Donnie had already been working on, and then just this last year, we went to the Parguera Fringe site, the Hobos Bay site, and then the Piniones Fringe and Backwater sites, and took cores, brought FIU, USF people to Puerto Rico, had 3 undergraduates from UPR working as part of an RU, and we brought everybody back up here, took samples in the South Florida areas, and then we brought everybody to USF so that they could see what Donnie does after crawling through this ridiculous jungle gym and then pounding a sledgehammer on top of a graduate student's hands so that he can get this PVC pipe deep enough. I mean, it's the most ridiculous thing I've ever seen. But the art is that Donnie brings these cores back, and then he does this lead-210 dating, which has a good horizon over the past 100 years. The limitation is it's only about 100 years. But anyhow, he can go through and he can estimate the sedimentation rates and how they've changed over a 100-year period. Well, that's a really powerful technique because we're now able to measure sea level rise pretty accurately. We're getting better and better. And so, we know that 3 years ago in South Florida, the sea level rise was maybe 2 millimeters. Last year, they upped that to 4 millimeters. This past year, they said 8 millimeters. Okay. So it's rising, and there's all kinds of dynamics you all know about. The flow patterns are changing. Some stuff is coming from the north that never did. The south flows are still coming, and so it's just, they're piling on South Florida, right? We got 2 flow paths that are hitting together and just causing this increase really quickly. But Donnie's methodologies allow us to say, well, there's no way, or yeah, the sedimentation rates basically are masking the sea level rates. And so this is the data we're looking for. I mean, here's sea level rise, here's accretion rates. If you stay here, you're going to live. If you're down here, you're going to go away, right? And now the next question is, if you're going away, is that simply that you get replaced by salt marsh or something, or do you basically start working backwards and moving more inland? And mangroves have a funny physiology. So this is the kind of stuff that we're doing now with Donnie and making some very interesting comparisons. That collaboration led to another collaboration with another person Randy Parkinson, who got his PhD at Erasmus. Randy's really interested in all these different methods that people were using, because different labs in different parts of the world try to measure sedimentation or accretion rates differently. So Donnie uses lead-210. Other people look at cesium, about the same time horizon. We certainly know when the atomic bombs were tested and all that stuff. And then there's lead C14. It allows you to go back longer in time, but it doesn't allow you to divide time. In other words, it's just a single integrator, right? You can get an average sedimentation rate over a few hundred or 1,000-year period, but I can't tell you what happened 4 years ago, whereas in these two methods you can. And now the interesting thing is people are always looking for an easy, repeatable, standard solution, right? So they now have this sets approach where they're pounding in these rods into permanent positions and then actually physically measuring sedimentation rates. And so now there's a question, you know, which of these are better? What do they tell you? And so this has started a whole new interesting collaboration. Well, we were in Puerto Rico in May and June doing these, all this sampling in the mangroves. But this is what Puerto Rico looks like now. So I happened to fly in a couple of days after the hurricane, and this is a drone flight over the El Yunque. And you can see that there are very, very few leaves left. It begs the question, what happens to sedimentation rates after an enormous storm like this, after an enormous disturbance? And you can, in fact, hypothesize that the amount of downed organic matter, which will be somewhere on the order of 3 kilograms per square meter. So think about that, 3 kilograms of wood and leaves per square meter throughout the forest floor. All that stuff has to go somewhere, right? Some of it will decompose in place, but a lot of it, when the next rains come, will start getting washed into these streams and get transported down. And we may have the single largest accretion rate a year that we have ever measured, right? So really, it's a very sad thing, sorry, a sad thing, but it gives us an amazing opportunity because we just measured all this stuff. We just made these estimates, and now we're going to be able to go back 1 year later and say, what happens after something like this? And it may be that ecosystems are incredibly resilient, that an event like this, which has a huge impact up here, all these trees will come back. You know, a lot of them, some individual stems will die, but in 10 years you won't know this ever happened. But this flushing of organic material may completely alter our notion of what annual sedimentation rates look like in coastal mangroves. If only Irma would have hit harder. That's the scientist in me. All right. So this is just down and personal. This is one of my long-term study streams. This is what it looked like when I was there in July, and now you can't even find it. You can't get into it. What happened there is just unprecedented. But you know, it gives us a great opportunity to try to understand all of this organic matter processing, nutrient cycling, etc. Okay, so we started with Everglades, we added mangroves. Our newest thing is shallow marine habitats, right? So it gets to the mangroves. Some of it gets taken up, some of it gets trapped, but lots of it probably goes off to the seagrass beds, the oyster beds, and the coral reefs. So that's our newest thing. We have started a little bit of stuff. In fact, the supplement that Adil was talking about is the University of Puerto Rico CREST looks at the same kinds of things, stressors. They include contaminants, but they also include things like acidification, temperature rises in water temperatures. They also look at rivers, mangroves, estuaries, but they spend a lot of their time and effort thinking about shallow marine habitats. But their responses are really interesting. They're looking at neurological impacts. So how neurological pathways are affected by these various stressors, how that alters behavior, how that might alter then predator-prey dynamics, and ultimately community dynamics. So it was a no-brainer to say, well, let's bring the Puerto Rican folks over to FIU for 6 months, let the students keep working on their stuff, and while we're here, let's talk about your guys' approaches and our guys' approaches and what we can do together. So this is our sort of newest thing. We do have, a lot of you know Jim Forkurian. Jason Howard is one of his PhD students soon to finish. He's been looking at sort of seagrasses as a carbon, blue carbon storage. So he's very interested in carbon dynamics in seagrasses. And I know I talked to somebody today and they were interested in the same kinds of things. So Jason has been sort of looking at CO2 and how things come from upland or inland hitting the seagrass beds affects CO2, as well as other things like acidification, changes in temperature, etc. And so he's been sort of also measuring sediment characteristics and looking at sort of the net carbon fluxes and trying to understand the importance. Of course, just like you guys, we undergo these periodic rapid seagrass bed die-offs, and then other times the seagrasses seem to be doing perfectly fine. Okay, so he's coming up, he's concentrating mostly on Florida Bay, but he's looking at sort of how the various stressors and environmental conditions impact seagrass. The interesting thing about Jason is he's a master brewer, and he's made a seagrass beer. It's not bad. It's not great. You know, if you feel sort of a physiological need for silica, you might. He really did make this beer. This is another one of our undergrads. One of the questions now in seagrass beds, and again in the world of blue carbon, is how fast does stuff like detrital decompose? And the problem is everybody has different methods. Some people use the teabag method. You can use your species-specific decomp methods directly. What she's interested in for her undergrad thesis is if she can come up with sort of a standard methodology that you could use anywhere under any conditions so that you could get truly comparative data. And so she's trying to come up with a way of sort of measuring this stuff uniformly. So she's come up with this sort of ingenious approach. She's using these little canvas strips, and they mimic seagrass leaves, or whatever you would call them. She puts them in, and as they decompose from microbial oxidation and microbial stuff, it sort of mimics the decomposition process. And so when you have fabric, what happens is you lose the tensile strength of the fabric as it becomes to decompose, right? And so then she can use some kind of a thing and measure how tensile this stuff is as a surrogate or a correlate for decomposition. And so then she can actually measure the weight loss, the actual true decomposition of this fabric, and she gets a really nice relationship. So it looks kind of promising, pretty amazing for an undergrad. To set these things out, go out, and very quickly you just pull on them and it says, oh, it's this number, put it through the regression, it says, oh, it's decomposed at this rate, right? So it's really quick and it's very uniform, right? Same strip, same stuff, and you can put it anywhere in any condition. So seems like a really interesting approach. Kevin Boswell is sort of our autonomous surface vessel guru. We have been working on this kind of a autonomous vessel that is equipped with side scanning sonar, radar, and then the thing I can never remember. Help me, somebody. We've been talking about it. The what is it? What? The acoustics. Yeah, all the acoustics. And so, and he's been doing some very cool stuff using sort of this multi-frequency wideband acoustics. It's got really fine resolution plankton. It's got a pretty good radius in terms of driving this thing and sampling. Not only does he get the food web that's in the water column, but he actually can get some, and he can certainly map and measure physical habitat. This is one of his scans, so he sees a crab trap, a 50-gallon drum, all this stuff that's been deposited on the seafloor. Of course, this is off-the-shelf technology. The art is having programming capabilities to harvest this amazing amount of data and make sense out of it, okay? And so that's what his sort of claim to fame is he's got an army of sort of programmers that are writing the next generation software to sort of take all these disparate data sets, put them together, and come up with a three-dimensional characteristic of what we have. I think one of the coolest things he did was he took all of the fish, so he actually did this data collection, but he actually caught the actual fish and he put them through an MRI machine, right? And the MRI machine allowed him to look at where the swim bladders were, the shape, the size, the tilt, where they were in the body cavity. And using that, he came up with a principal components analysis that allows him to identify fish to species through this remote sensing capability. So that's a pretty cool technology. It's a pretty cool technique, and it's proving to be pretty robust. The newest thing we're doing is, of course, it's one thing to do that in a shallow reef, an open water habitat. We know very little when we talk about that transition between the Everglades, the mangroves, and the near shore habitats. The mangroves are virtually impossible to sample if you want to look at fish. You can try to snorkel, but it gets cloudy, muddy, it's usually dark, it's very complex, you can't see. And so now what we're trying to do is come up with the programming to allow this kind of equipment to basically pick out fish, crabs, and invertebrates from the mangrove roots. And we're doing it experimentally right now. We're doing it in tanks about the size of this room, where we're stocking the tanks with a known community of fish, and then changing the complexity of the habitat, you know, in terms of sort of mangrove densities. And so far the data look pretty interesting. It looks like we're picking out a high proportion of the actual fish in the living invertebrate, things, and separating them out from the mangrove roots, and actually coming up with pretty much the right species composition based on his swim bladder stuff. But it's pretty early on. Okay, so at the far biological side of sort of what does it matter, how do these different things impact the communities, we've got a group of people, including people from public health, who are looking at epigenetic impacts of various stressors, okay? So it could be anything from copper, mercury, it could even be sea temperature, et cetera. And of course the idea here is that as organisms get stressed, they have this interaction between their genomes and the environment in which they basically block different pathways. And the more stressed an organism is, the more pathways they tend to decommission to save energy, right? And so you basically get these short-term impacts where something is so stressed it no longer grows, it no longer reproduces, it doesn't grow. Ultimately they die. But under low levels of stress, they knock off very subtle sort of enzymatic pathways and stuff. And so these guys have been looking for markers and looking at ways of using this as an early warning system that a system is stressed, again at very low concentrations. And so they're using biomarkers. They're using corals and bivalves as their two study organisms. And of course, we all know about the brevetoxins associated with red tides as an important stressor, certainly for bivalves. And basically, they're finding these very interesting results that very subtle stressors from brevetoxins and stuff are creating pretty major epigenetic responses, but most interesting is those are heritable. And so you get an epigenetic response where you perhaps slow down parts of the reproductive cycle, but then you pass that on to the next generation. And so it doesn't take long for a population modeler to say, wow, that's going to be a very quick decline in the population if that's a heritable epigenetic trait. So that's the kinds of things these guys have been looking at as, again, a low level stress, but a huge potential impact. One of the last things I want to say is you can't do this breadth of work and give students sort of the ability to not just be an academic. Lots of the students don't want to do what I did, get a bachelor's, a master's, PhD, and go work at a university. So we have this amazing group of partners who are part of the CREST, and they come to our annual meetings, and it's a great give and take. They say, well, these are the research questions we think are important from a management resource agency perspective. And so the students and these guys kind of pair up together. Sort of students get ideas about how to ask relevant questions based on what the management agencies are telling them. And then the management agencies are providing us with intern opportunities. So our students, if they choose, can go do an internship with one of our partners in the summer, still do some research, but actually learn what life would be like if this was the job that they took on, if that was the career path they chose. So we have this really great set of of partners. I want to just really quickly, this is our website. And so the real reason for me to come here and talk, besides to get to meet you and hear what all the wonderful things you're doing, is to think about possible collaborations, to think about ways we could strengthen sort of what each of us are doing, finding synergisms. And so we have lots of different opportunities here. I can see all kinds of new proposals that can be written from the people I've talked to, but we also have ways of exchanging students for summer programs, both ways. Everything I ever talk about is both ways. And so, you know, go to our website. I think if most of you, if you go to the opportunities part, that's where you'll find out, you know, if we're giving away money for certain kinds of equipment, if we're looking for people to come down and work for the summer, what classes we take. So one of the discussions we had this afternoon at lunch with the grad students is, you know, we're pretty close, technology is pretty good. It should be pretty easy to put together some very cool distributed seminar series and stuff like that. We have so much in common in terms of the things we're interested in. And so we really ought to be thinking about how to partner and create some of these sort of joint ventures. And then, so down here is sort of, for those of you who are interested in postdocs, PhDs, this is, you can go here and find, it's a really quick and easy application. I say that, I mean, of course you have to apply to the university and then to a department and find an advisor, but then you can say you want to be in CREST, right? So, and there's on the web page, there is a very good description of who we are, and who the faculty are. So if you're one of these hardcore chemists, then these are the people you want to talk to. These are all the faculty, and then there's another page of all the grad students. And of course, a smart grad student who's looking for a grad program goes to the other grad students in that program first and says, how is that? You know? And so we actually have another page that lists all the grad students and stuff like that. So I thank you an awful lot for your time, and I'm happy to have any discussion you would like.